So it's my pleasure to introduce Mark Fallon, and we're looking forward.
Oh, thank you. Like being a pop star, having a microphone like that. Right, so I guess I'm the warm-up act this morning, and I'm also the token bacteriologist here. So although I work on genomes, I work on bacterial genomes, which are far more interesting and far more complex in the round than the human genome. You know, people who work on human genome, you've got this single, fixed, simple target. We have riotous complexity to deal with. Now, I'm going to take you through diagnostic metagenomics and just a brief overview of the impact of high-throughput sequencing on my field, and then take you through some case studies looking at diagnostic metagenomics, and then just a little brief look backwards, 200 years to diagnosing the dead, a second look at diagnostic metagenomics in a modern context, and then some recent work I've done just in the last few weeks. I'm going to mention cloud computing briefly, and then look at what the future might hold. So for me, around the turn of the millennium was a very exciting time to be in microbiology. It even made me kind of wax lyrical, you know, you feel like you're in that Keats poem, like some watcher of the skies when a new planet swims into his ken. Basically what happened then was that we saw for the first time the inner workings on our computer screens of humankind's most fearful microbial adversaries, from the causative agent of the Black Death to the White Plague, tuberculosis. For the first time, we could actually get to grips with how these organisms function. And it was rather like when the Voyager probes went through the solar system and turned little smudges down a telescope into newly mapped worlds. We had the same kind of elation as we explored pathogen biology in this way. Then, of course, as the millennium rolled on, we saw steady improvements in sequencing. But then there was a kind of step change. So sequencing was keeping up with Moore's law, and then suddenly around 2007, foot went on the accelerator, and what's sometimes called hyper-Moore's law came in, where the advent of next generation or high-throughput sequencing came along. Initially this brought a fresh infusion of interest into the field, because you could actually sequence genomes in multiple centres. But then we even got to the stage where we got these so-called benchtop sequencers, where any average-sized university or even clinical lab could actually start doing sequencing for themselves. And in fact, we towards the end of last year, wrote a review, something like 20 years of bacterial genome sequencing. Now, one of the kind of highlights of my career in terms of bacterial genomics was looking at this outbreak. Some wag called it the sproutbreak in Germany. It was caused by bean sprouts. It was actually caused by this particular kind of E. coli. And we worked with collaborators in Germany and in China to get a genome sequenced. And we also had this crowdsourced effort where people from around the world, bioinformaticians, are all contributing to the analysis of the genome sequence. Now, I'm not going to dwell on this very much because I've told this story before. It's getting to be a bit of an old story now. But basically, that culminated in us getting a New England Journal paper. And we were very pleased with that. But we hadn't actually met the German collaborators in advance of the paper coming out. So a few weeks afterwards, we went over to Germany and had some champagne with them. And we said, what are we going to do next? You know, we've done well to get this paper in the New England Journal, but people will just say you're one-hit wonder, one-trick pony. Is there something we can do to show that we're actually better than that? And so we sat, there's me with Martin Epfelbacher, the guy in the middle there, and we were saying, what are our unique selling points? So I said, well, our unique selling point is we're quite good at bioinformatics and genome analysis and that sort of stuff. And he said, well, his unique selling point was he had a freezer full of shit, basically. He had over 200 samples, fecal samples from the outbreak. And, you know, almost in the words of a rap song, the project became, let's sequence the shit. We said, why don't we just do that and see where we go? Now, obviously, there's a more serious rationale for this, because if you look at where diagnostic bacteriology is currently sitting, it's sitting in a 19th century groove. So we're using technologies, relying on technologies that were invented in the 1880s. So Graham showed us how to do gram staining, which is still done today. And Robert Koch was the first person to say, well, actually, you can grow these bacteria on solid media, and you can then pick individual colonies and propagate them in pure culture. And those two approaches still inform most of bacteriology to this day. Virologists are a bit smarter, they've been using molecular methods for a bit longer, but bacteriologists still rely on this. And we think, well, yeah, in the 21st century, can't we actually come up with some kind of step change and actually get rid of these approaches? And so what I propose is that we do shotgun metagenomics on these stool samples, and that we actually try and develop a technique of diagnostic metagenomics. So in the initial paper in the New England Journal, we went from stool sample to the organism being cultured in pure culture, and then genome sequence. Could we not go directly from the stool sample to the sequences and get the same kind of result? So when you're doing this, you start off with a complex mixture of organisms, you end up with a complex mixture of DNA from different sources, and then all the heavy lifting is done in the bioinformatics stage. And this is in contrast with conventional bacteriology, where actually an awful lot of work goes into growing organisms in the first place. So you have many different atmospheres, many different culture media, very complex workflows. It's very simple. You just extract the DNA, sequence it, and then the hard work comes later. So they sent us 20 samples to start with, and the very first sample we looked at, we were very much encouraged. I mean, I started out thinking, well, if we can actually detect the sugar toxin gene, which is the most important virulence factor of the strain, in these samples, that would be an outcome worth having. I never imagined what we actually got, though. So when we aligned the reads from the metagenome in this particular stool sample against the E. coli outbreak strain, these are coordinates of the outbreak strain genome in millions, we basically got around 20-fold coverage of the genome, just out the box. And we get this nice kind of smile, which is something we'd expect to see, because when you have a fast-growing cell, you get a slight over-representation of the regions around the origin of replication. We also were able to detect the sugar toxin gene easily enough, and around the area where the sugar toxin gene was encoded, there's a little upswing. And what we took this to mean was that there's actually a bacterial virus, a bacteriophage, actually encodes the toxin, and that bacteriophage can sit in the E. coli genome for most of the time, but then it can also be liberated as the phage propagates and become these free phage particles. And we think that we are detecting here those phage particles, the sequences from those. And we were able to use a technique called MLST, multi-locus sequence typing, which is an established technology where you just sequence 7 housekeeping genes. We're able to apply that technology, that approach to this genome recovered from the metagenome and actually come up with a sequence type for the strain. So that was actually quite rewarding. We're very pleased with that. And it's one of an example where, time and again, I've been surprised by how successful experiments have been. And now that sequencing has become so straightforward and so easy, it's often worth let's just sequence it and see what happens, is, I think, a very useful approach, because you can be surprised by what you're seeing. Now, we tried to work this up for publication in JAMA, the Journal of the American Medical Association, and because they were having a special issue on genomic medicine, and we said, well, you know, genomic medicine is not just about human genome, can we do something about bacteria? And the editor said, yeah, okay. And then we discussed what we were going to present, and he then spoke to his own friendly expert. Turned out to be someone I know, David Relman, who said, well, you know, basically what these guys are doing, they're just aligning their metagenomes against the genome they know, because they've already got that knowledge from their previous work. But it would be far more interesting if they could actually do this de novo, so they could actually solve the problem without knowing what the genome looked like in the first place, without even knowing what the pathogen was in the first place, because that would then prove that this kind of approach might work in an outbreak setting in real time. And it's a bit like, you know, it's the difference between solving a jigsaw when you've got the lid, and you can look at all the pieces and where they go easily and put them together, versus not knowing what your jigsaw is supposed to look like at the end. And so, okay, that's a hard ask, but we'll take the challenge. And Nick Lohman, who was working for me at the time doing a PhD, he took up the challenge as a bioinformatician. And he first approached the issue by saying, well, let's have a look at the contigs, environmental gene tags that we call them, that were present in at least 2 of these fecal samples. So we ended up with 40 samples now from the outbreak. And here he's just tried to project the taxonomic complexity in the gut microbiota, in the gut microbiome of these patients. So what he's done is on one axis he's shown depth of coverage. On the other, he's used GC content, which is a handy way of just separating out bacteria according to their taxonomic status. And then he's done a rough and ready taxonomic assignment to the reads that he's got. And you can see that when you look at these, it's a right old mess. And the idea that you could ever drill down through that lot to find a single causative organism in an outbreak is quite challenging. So Nick said, well, what simplifying assumptions could he bring to bear on this? And the first one was that he said, well, if this is an outbreak, and we've got 40 samples from the outbreak, let's assume that we can at least in half of these samples capture the sequences from the outbreak strain. So anything that's not present in half the samples we can throw away. And when he did that, it did simplify the taxonomic landscape quite considerably. And you can see this pinkish stuff here, which is the Enterobacteriales, where E. coli sits, starts to become more prominent, but there's still an awful lot of other stuff there. So he scratched his head again and said, well, what else can we do to try and home in on what's causing disease here? And then he's a very inspired individual. He came up with an idea. He said, well, why don't we just subtract what's in normal people? And obviously, it would have been a bit onerous if we had to go and collect stool samples from 40 age-matched Germans wandering around Hamburg. But luckily, he didn't have to do that because there are a couple of projects, one in particular in the EU called MetaHit, where people had already been doing that and they'd already put this information into the public domain. So they went and pulled down 40 samples that he could match as best as he could and subtracted anything that was in those samples from what he found in the samples from the outbreak. And then he ended up with a pretty much first approximation of the E. coli genome from the outbreak strain. If you're savvy to these kind of things, awake in the morning, you'll realise actually what he got was he subtracted out the core genome of E. coli as well, because everyone is carrying E. coli. But he managed to capture the strain-specific sequences, and then with help from another guy, Chris Quincy, he was able to do some interesting other work to actually get the whole E. coli genome back out again. But the end result was we got our paper in JAMA, we got the genome reconstructed from the metagenomes of these stool samples without having to culture the organism first. So that was a nice result. Then what I did a little bit later was we went and looked at some lung tissue, 200-year-old lung tissue. And I'm not going to say much about this. This is from a series of mummified corpses from Hungary from the 18th century. And we were able to get TB genomes out of this. Now, as I'm here talking to a clinical audience, I'm not going to spend much time on that. If you're interested, you can look at the paper. And in fact, the American Society for Microbiology have made an interesting little app. There's the URL for it there, where you can actually go and explore the work we did on this interactive kind of landscape, where you can go into a museum and see the stuff and so on. But again, this was a kind of launch point for further questions, just like in the previous thing. So this work actually caught the attention of Bill Gates, and he tweeted on it. And that gave me bragging rights at home, because my children said, oh, Bill Gates knows about Dad's work. You know, that was really interesting. But the problem is, of course, this is diagnosis in the long dead. What's the point in being able to get TB genome sequences out of people who are 200 years old? It's not really going to help you very much in the clinical arena. And so there's a phrase that you sometimes see in the dissecting room, mortui vivos docent, which means that the dead will teach the living. And we said, well, actually, has anyone done this on contemporary sputum samples from TB patients? No. Why don't we try that? And see if we can replicate what we've done on these mummified tissues on contemporary sputum samples, and that's exactly what we did. So with collaborators in West Africa and the Gambia, my PhD student Emma Doughty, shown here, was able to collect lots of sputum samples, which is probably second only to collecting stool samples as a most disgusting kind of PhD project to do. She got a large number of these sputum samples, extracted DNA from them, and was able to then sequence the DNA. She got various results. One of the problems when you're doing these kind of things, and it was something we thought would be a problem with the stool samples, but turned out not to be such a problem. We did think it was going to be a problem here, and it was to some degree, which is that obviously you've got lots of human DNA there. So if you're looking for the microbial sequences, you have to do that in the background. And so in some cases, you've got over 97% of the sequences were actually aligning to the human genome, and we weren't interested in those. But then when she aligned against this H37RV, which is often taken as the reference genome for Microbiome tuberculosis, she still got sequences. We got different levels of coverage. The best we got was about one-fold coverage. Actually, when we then went more stringently, it came out to about 0.669-fold coverage, which, considering it's coming straight out of sputum, is still not too bad. And the thing is that the number of bases we're still getting tens of thousands or hundreds of thousands, or in one case millions of bases of TB. So that clearly gives us a diagnostic signal. This is, without any doubt, Microbiome tuberculosis in these samples. That's plenty enough to tell us that. But the other thing was that when we get those levels of coverage, we were even able to map the TB sequences onto a phylogeny of TB strains and say what kind of TB strain was present in the sample. So we were able to say that in some cases they belong to this lineage called Lamb, the T lineage, the Harlan lineage. And in two cases there was a particular lineage that's found only in West Africa, so-called West African 2 or Microbiome africanum lineage. So this was an interesting finding that we could actually go beyond just getting a diagnosis, but also do some kind of epidemiological typing. Now, I won't oversell this because the one thing we weren't able to do was to get enough coverage to call out SNPs that might be associated with resistance. So we couldn't tell whether we were looking at sensitive or resistant microbiome tuberculosis using this approach. But as with the previous study, we were pleased that it actually worked out the box fairly well, and that was very promising. And Emma has gone back now and is trying to improve the technologies, the approaches, and see if we can get rid of some of the human DNA and increase the proportion of microbial reads so that we might get deeper coverage in the future. Now, the last thing I'm going to talk about is something that's been on my mind in the last couple of years, and particularly in the last few months, and that's looking at patients in the intensive care unit. And when we have a patient goes into the intensive care unit, they will have started off, at least before they went to hospital even, with a healthy microbiome in their gut, but over time that becomes sick. There's an attenuation of the microbiome in terms of complexity. It gets simplified, largely as a result, but probably not entirely as a result of antibiotic selective pressure, because all of these patients pretty much getting antibiotics at some stage in the intensive care unit. And the problem there is that this very much simplified microbiota loses the property of colonization resistance. So it can no longer see off any invaders that might be coming in to try and take over that particular niche. And what you get is colonization with multi-drug resistant pathogens, which then proliferate and in the fullness of time then spill over, say into the bloodstream, or start to cause ventilator-associated pneumonia, cause line infections and so forth. They start off by proliferating in this particular niche. And my interest is, well, is there a way in which, given that so much interest in combating antimicrobial resistance at the moment, that we can come up with an ecological understanding and ecological interventions here, so that if we could restore that sick microbiome back to something that's healthy, then we could actually stop these nasty organisms getting a foothold in the first place. And as we mentioned yesterday, one option would be to do fecal microbiome transplants. The thing is, though, that if you look at the literature, there isn't very much literature on documenting this process, and certainly not using the most up-to-date methods of sequencing and metagenomic sequencing. So just in the last few weeks, I got 10 samples from the intensive care unit, just random samples, and said that just send me the stool samples. We don't have to get ethics if we don't know anything about the patient. Just send me some stool samples from intensive care unit patients. And what I did was I then ran them through a commonly used pipeline called Kraken, which tells you the proportion of reads belonging to each particular taxon in your metagenome. I was quite astonished to find that in several of these here we have clearly what would be considered potential pathogens. So we've got 3 of them, we've got Enterococcus faecium in them. We've got Klebsiella pneumoniae, E. coli as well. But one of the most striking things was that the proportion of the reads that actually belonged to this pathogen was really quite impressive. So one there, we've got over 50% of the sequence reads coming out of this stool metagenome belonging to this one organism. So this confirmed what we'd seen earlier. So I didn't actually say, with the E. coli outbreak, what we realised was that in a disease state, things are quite different from in a normal state in terms of the proportion of organisms and the number of different organisms, but also whether one is predominant or not. So in normal individuals, E. coli is very much a minor component. So the Bacteroidetes and the Firmicutes are the 2 phyla that predominate in normal people. But we'd seen in the German outbreak that E. coli became predominant and we were able to detect it. And the same thing is going on here, that Enterococcus faecium in several of these samples has become the predominant organism in this particular environment, such that it really has just taken over and accounts for most of the reads. And in fact, I took the 3 that had Enterococcus faecium and then just ran it through a prediction pipeline, which is usually applied to cultured organisms, to see if we could work out what the type was, sequence type of it was. So we could show that they were different sequence types, and therefore there wasn't any evidence of cross-infection. I didn't know anything about the context of these patients. Maybe they were in adjacent beds at the same time, maybe they weren't. In fact, there was no evidence of cross-infection. These were actually different strains of Enterococcus faecium. But we could also detect that they had vancomycin resistance. So we could go directly from the stool sample to actually being able to say that there's a nasty organism, but it's also multi-drug resistant as well, very quickly. And again, this is very preliminary. This is just out the box. It's not in any way a polished approach, and that's what we've got to devise in the future. But it's been very interesting and reassuring to see that we can get these things working. So I used another approach where we just took this multi-locus sequencing approach, typing approach, and applied it to these metagenomes. And in fact, it pulled out various alleles from different organisms that were in these databases. And in a couple of them showed us things that we didn't see when we first looked, like Candida albicans were also present there. So in conclusion, it's clear that when you use these sequence-based approaches that you can see that in the critically ill patient, their microbiome is very sick, is the predominant thing. And the pathogens bloom in this environment, and that because of that, because it's a very simple microbiota, you can actually just use this diagnostic metagenomics approach and get useful diagnostically, clinically actionable information just by sequencing the samples. Now, in conclusion, I'm not the only one using these approaches. Shortly after our paper in JAMA, there was a paper came out in the New England Journal where they just sequenced some clinical material and were able to diagnose neuroleptospirosis in a patient who was really quite ill and nobody knew what was the problem, and they did it with sequencing. Nick Lohman, after he left my group, went on to do some very exciting work on Ebola genome sequencing using this portable device, the MinION, out there in West Africa. And just in the last week or two, a paper's come out from a group in Norwich showing that you can actually use this kind of approach on urines. Just sequence the DNA you get out of a urine sample, and you can identify the pathogens, you can call out the antibiotic resistance genes. So this is clearly an area which is going in an interesting direction. It's worth stressing that there are some pitfalls here. There's something that we call the kitome. So this paper here came out earlier this year, and this is the only paper I've been able to find which speaks directly to the topic of this here, which is looking at what's circulating in the blood. So these people were looking at sequencing organisms that were causing fever, and they did find in this paper some true positives. So they had a patient who had an overwhelming sepsis. They were able to detect E. coli sequences in his blood, and there were a couple where they had one with scrub typhus, the other one with dengue, where they're able to get sequence-based information directly from the blood without having to do anything else. But the problem they got, though, is that when you're looking at, in this context, the amount of DNA that comes from the pathogens and the amount of biomass of the pathogens that's present in the sample is much, much smaller than it is, say, in a stool sample. And therefore, your risk of actually getting false positives, contaminants, is much greater. So they basically got a kitchen sink's worth of contaminants. They were very honest. They presented 3 separate tables of sequences from organisms where they got sequences in every sample they looked at. And they wisely, they didn't over-interpret their data and say, well, these are present in everyone. They said that these must be contaminants. It seems that many of these are coming actually from the kit components that Illumina supply us with. And the same problems arise in many different contexts. So there's a paper that came out a year or two ago from some former colleagues of mine looking at this issue and saying that actually many, many papers, dozens of papers out there, people have naively said, oh, we found this in this sample. So you try and look at somewhere like the surface of the conjunctiva where you don't expect there to be any bacteria, you find something, but what you find is the contaminants, and there's all sorts of things out there. So actually I wrote a review and then Nick tweeted on it a few weeks ago saying a potential concern with the ease of these technologies and the growth of these technologies is that you've got the growth of a wild frontier of microbial genome and microbiome analysis performed by non-experts, hand-cranking data through pipelines they do not fully understand, and then naively interpreting results without engaging the healthy skepticism of the seasoned expert. I was in full rant mode when I wrote that. Eternal vigilance is likely to be the price to contain the equivalent of microbial genomic astrology. And this is a problem that we have here, that you have to be very careful in interpreting the results and contextualizing the results. In addition to the kit contamination, there is actually carryover from one run on an Illumina instrument to the next. And you have to be aware of that as well and be very careful about that as well. So, just before I finish, I give a shout out here for a project I'm involved in. So, it's all very well generating all the sequence data, but you have to have the wherewithal to analyse it in terms of the bioinformatics capability. And we have this project called CLIMB, Cloud Infrastructure for Microbial Bioinformatics, which is an £8.4 million funded project from the MRC in 5 universities across the UK, where we are providing end users with bioinformatics resources. We're using this route of virtualization, so they get virtual machines that they can log on, they can create and then log on to. We're providing training. We've got research fellows and space, and you can find out more about the project here. We've got a place called Bryn where you can, if anyone's interested in doing work on microbial or pathogens, you can go and launch your own virtual machines, and then there's plenty of help and discussion on the discourse site. So why is this relevant to a meeting like this? Well, one of the interesting developments is that we are all converging in the same kind of direction in different branches of laboratory medicine. So if you went back 20 years, a clinical chemist, a histopathologist, a bacteriologist might not have much to talk to each other about because they're all using completely different approaches, looking in completely different directions. But in recent years, we're now converging on the fact that isn't it interesting to look at macromolecules, informational macromolecules in tissue samples, and squeeze as much information out of that as you possibly can. So this can be in shed DNA in urine and feces, but obviously circulating DNA as well. And the big hope is that the technologies like the nanopore will make this very much more accessible. And there is even talk that this kind of nanopore sequencing might work for RNA and for proteins, so that you might be able to get more information than just from DNA sequences from these approaches. And obviously this is applicable to all of these different areas, so we can look at the germline genome, somatic genome. We can look at microbiomes and we can look at infection, but we can also use these kind of approaches to track inflammation, cancer, fetus, transplants. People even saying that maybe you can look at the state of someone's atheroma from looking at circulating biomarkers in this way. Now, of course, it's difficult to make predictions about the future. Someone pointed out to me that 25 years ago, I said, oh, the whole of bacteriology will be swept away by PCR. And it didn't quite happen like that. So whether in another 25 years we'll have swept away culture microscopy and culture, and everything will be done by sequencing? I don't know. When I first gave this talk, I gave it in Berlin, very close to where the Berlin Wall was, and I put this quote up, "I've seen the future and it works," from an American journalist called Lincoln Steffens, who in the 1920s went to the Soviet Union and came back and said that, said he'd seen the future and it works. Obviously the Soviet Union did work for another 50 years or so, 50 or 60 years, it eventually failed, so maybe not. The other thing is, of course, and I think it's a salient point that was discussed yesterday, is that actually getting these things to work in an academic context, when you're in a centre of excellence and you've got plenty of money and you've got all these very clever people around, is one thing. Getting it to work in a routine clinical context is quite another. And this quote here from the cyberpunk author, the future's already here, it's not very evenly distributed. I think it's very pertinent at that point, that it's not enough to just do a thing once and show it works. You have to then get it out there into practice. And then finally, predictions are difficult, especially about the future. And the provenance of that quote, I haven't been able to track down. Yogi Berra is one individual. I think the Simpsons have used it as well. Anyway, that's me finished. I'd just like to acknowledge all the people who've helped me over the last few years. Thank you very much.
I guess there are loads of questions, so I hope you can answer at least a few of them.
So when you were doing the E. coli outbreak analysis, it looks like your best win was removing stuff you expected,
Yeah.
then scaffold build and the rest.
Yeah, that's right. Yeah.
Naively, it would be easier to just scaffold build everything. Was it just that there was too few beats to do that?
Well, we did an assembly, so we weren't just looking at the raw reads. We did an assembly first, and then we then worked on contigs and removed contigs that had hits. It is fairly challenging to do assemblies on these metagenomes. It takes time computationally, but that was the route we took. I mean, the programs that most people use as their first pass actually just look at k-mers and actually work out what's in the sample just by looking at the reads themselves, and you don't have to do an assembly. But we did take an assembly-based approach.
Because it would be quite interesting to see what's new.
Well, the other issue is that, yeah, we're now, the Human Microbiome Project and other projects, we are getting progressively more and more genome sequences from all the different species that might be found in these samples, and many of the different strain lineages. And there is an argument to say, well, could you not actually align against all the genomes that are available and see which ones give the best hits? There is a program called Metaflan that does something a bit like that. It takes what it considers to be lineage-specific sequences and gives you a quick answer that thing's there or not. But the technologies, the bioinformatics approaches still need to be improved for this to work really slickly.
So what do you pick up except bacteria? So there must be some viruses or others.
So in these most recent things I've done with the ICU, we didn't actually look for viruses. One of the problems is that these quick programs that I mentioned, Kraken, that just look at k-mers, they depend on a library of organisms that they've pre-compiled, and the default is to just get bacteria. So it doesn't give you viruses, and it doesn't give you fungi, and you have to go back and it'd be interesting to do that and actually go back and see, well, actually, are there any viruses there? People in Oxford are actually looking at this kind of approach for norovirus, and you can get norovirus genome sequences out of stools using these kind of approaches.
I find there are so many hits to huge numbers of
Yeah.
viral genomes in the human genome.
Yeah.
We do that routinely in our own samples, and we find, there's just so much noise.
Yeah, the so-called microbial dark matter that, a lot of the reads that you get, you can't align them to anything, you can't assign them to anything. What are they artifacts, or are they things like, complex viruses that we just haven't actually got around to sequencing yet? I mean, the other thing that you see is 5X174 comes up because it's very commonly used as a kind of filler on the Illumina platform, and probably the the whole Illumina environment gets contaminated with Phi-X. And so that paper I mentioned, that they actually said, well, they found that. They found quite a high proportion of their reads were Phi-X.
This is probably a limitation for the sensitivity of this like shotgun experience, isn't it?
Yeah,
Because
it is.
I remember we once in pediatric oncology neutropenic fever is like the most common complication, and you do blood cultures before you start antibiotics, but you wouldn't let them become so ill that you really have positive blood cultures. And we tried by sequencing, and you got all this background information, but not yet the pathogen.
I know, and that is the problem. I mean, a lot of the time it's that actually septic, it's not that they've got an infection, it's that those inflammatory processes have been set in motion without the bacterium being there. In other cases, it is a problem that the bacterium or the virus or fungus is there in such small amounts that you're hard pressed to find it. In that paper that I mentioned, they did find, I think, 11 reads that aligned against dengue virus in someone's blood. Now, if you never sequenced dengue virus and you've never done any work like this before, how else would you explain those reads? They asked, that's even with 11 reads, it's still a result that means something. But it is hard. And I think there is going to be this interesting debate as to whether you go with shotgun metagenomics, which gives you the open-endedness that you might find something you're not expecting, versus trying to either capture things or culture things as an intermediate step before you actually go and do the sequencing.
More questions? No, thanks
Okay.
again.
Great, thank you.